All right, we're on the home stretch now of why things are so complicated. The sixth and final aspect of what makes the relationship between genes and traits complicated is the fact that genes can be turned on or off, or even kind of amped up or down by a whole range of factors. So even though you have a specific gene, a specific allele for a specific gene, it doesn't necessarily mean that you are actually going to use it. It might be turned off for a long period of time and then turned on, or might be on for a while and then kind of dialed down, and it won't make as much of that protein. So whether a gene actually ends up being transcribed and translated is due to a whole host of factors. And the study of those is called epigenetics. So when we're thinking about what regulates gene expression, what decides if a gene actually ends up being expressed, the protein ends up getting made, is the field of epigenetics. And it is one of the fastest growing fields in biology these days. So it's pretty cool. You already kind of know this, that some genes get turned on or off from the lab, right? We learned that in X inactivation, whole chromosomes, like the entire X, one of a female's entire X chromosome kind of gets condensed permanently during fetal development, and that X chromosome never gets to do its job. It's the other one, right? And that's what explained why cats who are heterozygous for the orange-black gene have both if they're female, because some of their cells silence the orange gene and some of them silence the black gene, resulting in kind of a patchwork effect of black and orange. So you're already kind of aware that some genes can be, or in this case, an entire chromosome can be turned on or off. Let's explore this some more. Your cells in your body are a wide range of shapes, functions. Here's some images, symbolize some actual photographs of bone cells. Look very different from neurons, which look very different from muscle cells. They have incredibly different structures, incredibly different functions, and yet, except for your sex cells, except for your eggs and sperm, let's set those aside for a moment, except for those, they all have the exact same DNA. They all have the same 23 pairs of chromosomes. So, what? It's all because some genes are turned off and some genes are turned on in each of these, enabling them to make some proteins and not make others that affect their structure and their function. Even for cells, let's say a cell in your digestive tract, let's think of lactase again. That's what the LCT gene means, the gene that makes lactase. You aren't necessarily making lactase all the time. You're making it when your body says, hey, there's some lactose here, right? So dialing up and dialing down when that gene actually gets expressed and makes lactase has factors that regulate it at pretty much every step. There are factors that regulate whether it gets transcribed. And how much. There are factors that relate to RNA splicing, which you can learn about in AP Bio or in college. A whole other little step that involves chopping up and splicing the mRNA once it gets made. Fun times. There are factors that relate to whether that mRNA actually gets out of the nucleus. There are factors that relate to whether that mRNA and how much of it actually gets translated. And even if it gets translated, then there are other factors that relate to, well, do we actually still need it? If we don't, we'll just stop it there. We won't export it. And we'll break it down. We'll break down those amino acids and use them for something else that we do need. So every step of the way, there are factors that can calibrate, do you or don't you actually use the gene? Does the gene actually end up doing its job? More recently, we've learned some really freaking cool stuff about how the environment plays a role in some genes that we thought were kind of all the time on or all the time off. So we're going to, within this video, we're going to watch another little video right now.
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
So as we learn about epigenetics, it kind of brings us back full circle to where we started, which was noting that both genes and environment play a role in traits, and seeing that that's kind of ground zero for how they interplay with each other, how environmental factors, like the video said, stress and diet and exercise, it's one of the mechanisms that those experiences, those factors can actually affect your genes. Not changing the code, but regulating which genes actually get transcribed and translated and which ones don't, or how much they do and how much they don't. Which is, I think, a healthy reminder for all of us that healthy living is important. Managing stress, eating well, getting enough sleep. These are all things that you already know can affect your health, but they can also affect how your genes work. And so I'll leave you with that thought of the importance of taking good care of yourself.